I'm here from the Center for Integrative Research on Childhood Leukemia and the Environment, and the acronym that I'll be using during the presentation is CIRCLE, because everybody needs an acronym. And the parent study for that center is the California Childhood Leukemia Study. So I'll probably at times lapse in between those two descriptions. So the topics that I want to cover today are these five. I want to talk about how the California Childhood Leukemia Study has measured multiple environmental exposures, and talk about some of the data sources that we have available to us. And then I want to talk about how we can systematically analyze these multiple exposures that we've collected. And then CIRCLE has two projects that could potentially lead to novel biomarkers of effect, cumulative effects of multiple exposures. So those two opportunities are to measure human serum albumin adducts, which is something that Martin talked about earlier today, as markers of multiple exposures, and also to talk about DNA methylation as a potential marker for multiple exposures. And finally, I want to talk about leukemia incidence in the Hispanic population in California, and I want to sort of view that as an opportunity to assess multiple environmental exposures and genetic sensitivity. So the first topic, measuring multiple environmental exposures. So the California Childhood Leukemia Study is a case control study that was initiated in 1995. It's a large study with approximately 1,000 cases and 1,000 controls. It's still ongoing. And we measure exposures in a lot of different ways. We use questionnaires and interviews to get a lot of information. And then we do some environmental sampling as well, air sampling and also dust sampling. Dust sampling and dust analysis has kind of been my specialty so far. We also do spatial analysis with GIS tools, and we do some traditional biomarkers in urine and blood, and we're also doing those two new projects with the Adductomics project and also methylation. So to go into a little more detail here, these are the various things that we measure with each one of those types of assessment. So the questionnaires, we ask about smoking habits, we ask about pesticide use inside the home and outside of the home, drug use, occupational history of the parents, diet, and various other health characterizations that the parents give us. The dust sampling, which is near and dear to my heart, as I said, we measure insecticides, herbicides, PAHs, PCBs, PBDs, heavy metals, and some markers of tobacco, including nicotine. So I'm going to talk a little bit more about those categories as sort of a model for how we can systematically assess multiple exposures. So we have all of this environmental data that we've collected, but to date we've sort of done a piecemeal analysis of all these different environmental exposures. So we're taking one exposure at a time and doing a risk analysis for leukemia, or we're doing possibly a set of environmental exposures like PCBs as a group, but still not considering how PCBs might interplay with other environmental exposures, chemical or otherwise. And it becomes sort of a data reduction problem because even though I'm not going to talk about the sort of things that Martin was talking about where we have omic style results, we still, with just our questionnaires and our dust assays, have generated enough data where we need to come up with some way of reducing it before we can start looking at co-exposures or multiple exposures and cumulative effects. So, of course, the genomics and genetic world has a whole series of tools that is tailored for data reduction or for basically visualizing multifactorial data. So we can use these same sort of tools, things like Manhattan plots and haplotype maps, or ways of looking at co-exposures for the environmental world in the same way the geneticists do in the genomic world. And this is something that's basically been pioneered by Chirag Patel. And one of the papers that he has published is this environment-wide association study, or EWAS, to be analogous to the GWAS, on type 2 diabetes. So here is my version of the tools that geneticists have been using for environmental factors. So this is looking at correlation of all the different chemicals that we've measured in our dust samples so far. So there's a list of 74 different chemicals, and they're grouped in the groups that I discussed on the previous slide. And the color bands on the outside of the matrix there are indicating the different groups. So it's probably too small to read, but it goes organochlorine insecticides, organophosphate pesticides, and then there's groups of herbicides. When we get to the second purple, it's polycyclic aromatic hydrocarbons, and then PCBs, polychlorinated biphenyls, heavy metals, and then the last band there is tobacco markers. The color scheme for the matrix is essentially that if it's red, that means that there's a lot of correlation between those two environmental factors in the dust. And if it's blue, that means there's an inverse correlation between the two environmental factors. There's a red line down the middle, which is just the one-to-one line there. But you can see there's also some regions of very bright red, like here and here. Those are homolog groups, so things like permethrin, the cis and trans homologs are really highly correlated with each other. But there's some a little bit more subtle groups that are potentially more interesting. So, for example, there's this group of light pink here, which is all of the PAHs correlated together. And there's a small triangle of pink here, which is the PCBs correlated together. And a similar triangle here for the heavy metals. But if you look at the whole area together, there's sort of a lighter pink outline there, where all of these things have some correlation. Not all of them, but there's overlap, and some of these things from these different groups are correlated with each other. And there's another interesting pink block up here in the OC insecticides, where they're correlated with that same group of other chemicals, the PAHs, PCBs, and the heavy metals. So you could take like a slice of this matrix and see how it how one of the chemicals correlates with everything else in the matrix. So for example, I'm going to look at lead because it seemed to have a lot of pink along that one row. So this is the extracted slice. And then there's all these different groups of chemicals that are correlated with lead. Some of them I guess you could have predicted, like the other metals. So things like nickel and copper you might have expected would be pretty highly correlated with lead. But there's also these other things, organochlorine insecticides, PAHs, and PCBs that we saw in those pink areas. And what's probably behind this is the fact that all of these things are also correlated with the age of the residence where we collect the dust. So especially things like DDT, DDE, the PCBs that have been banned for decades now are things that are going to be more prevalent in older homes. But also things that are still being produced, like PAHs, those are products of combustion, so they're always being generated. Those also are related to housing age and sort of the age of the dust in a home. So they are persistent chemicals that accumulate in dust over time. So I guess this is just a way to visualize our data and suggest that we could possibly identify some co-exposures that we could then use to investigate multiple exposures and the cumulative impact that they might have on childhood leukemia. And this is just with dust at this point, but we could expand it and include all of our questionnaire data and our data from biomarkers as well. So this is the Manhattan plot that's sort of analogous to the GWAS results that you'll see. Along the bottom are the same categories, and this is the dust-wide association study. Instead of the GWAS or the EWAS, it's a DWAS. I mean, it's kind of silly, but you see the vertical axis there is the p-values in a log scale, and the red line that goes across the plot is alpha equals 0.05, which of course a geneticist would laugh at. We don't see a lot of action there. There's not a lot of top hits, but it is an interesting way to visualize the data and sort of get a sense for whether these exposures are acting together in concert, if they're all associated as a risk factor together. PCBs is a risk factor that we've reported before. There's a couple here that are approaching significance. And all of these Dots are basically an odds ratio from a logistic model, and they're all adjusted for the child's age, sex, ethnicity, and the family's income. So again, this is just a tool that we might use to visualize our multifactorial exposure data. So now I'm going to talk about human serum albumin adducts. Adducts are addition products, and talk about how they could be used as markers of multiple exposures. So Martin gave me a nice primer earlier this morning. Human serum albumin is an abundant protein in the blood, and it has a long residence time in the blood. There is one free cysteine residue in human serum albumin that attracts electrophiles. So it attracts xenobiotic chemicals, but also endogenous things, including, label on here, reactive endogenous species. And we can measure these adducts of human serum albumin in blood spots from newborns that have been archived in California. And then we would have some measure of the child's exposure to reactive chemicals during the last month of gestation. So you could use these human serum albumin adducts in an exposome-wide association study. So the way that this was set up is in the figure here. You have a case population and a control population. You collect blood from each of them. You get your human serum albumin, which has some proteins that have adducts attached. You first need to isolate the adducted human serum albumin and get rid of the human serum albumin that does not have any adducts. Then you break up those adducted human serum albumins into peptides, and you isolate the peptides that have the cis34 residue, which is the one we're interested in, the one that has the ability to scavenge electrophiles. And then you compare the profile of adducts that you get at that residue from the cases to the controls in a quantitative comparison. And as Martin explained this morning, it's an untargeted or agnostic approach. So you don't have to have an a priori chemical that you're looking at in order to do this sort of comparison. You can just look and see what's different, and then you can go back and identify what the adducts were and what potentially the chemicals were and the sources of those chemicals could be. But you could also, rather than identifying specific chemicals, you could use it as sort of a non-specific marker of something like reactive oxygen species. And I'm going to talk a little bit more about that. So here is one possible mechanism for carcinogenesis that is involving reactive oxygen species. So it starts with carcinogen. It could be a chemical agent, or it could be, I guess, something like stress. And that carcinogen induces reactive oxygen species in the body, which creates DNA damage, genomic instability, and then that leads to cancer development and progression. So we have already measured some of these things in blue, right? We know what some of the potential carcinogens might be, all these things that we've measured in the dust and all these things that we're trying to assess by questionnaire or other means. And some of these things might generate reactive oxygen species. So they could generate all kinds of different reactive oxygen species. And it's not going to be specific for this assay. So what we're going to try to do is, again, use this residue in human serum albumin that will react with any of these reactive oxygen species and form a non-specific adduct. Now, human serum albumin is important because it's going to scavenge 80% of the reactive oxygen species that exist in the cytosol. So it's going to do a great job of telling you, it's going to be a great indicator of how much reactive oxygen species are present. So it's a potential biomarker of the cumulative exposure to all reactive oxygen species. Okay, so that is project 2 of the CIRCLE grant. Project 3 deals with DNA methylation, and we can also look at DNA methylation as a possible biomarker of multiple exposures. So I'm far from an expert at DNA methylation, but I think this is a nice quote that is a nice analogy as an introduction to what DNA methylation is. So if DNA sequence of the genome is like the musical score of a symphony, then the epigenome is like the key signatures, phrasing, and dynamics that show how the notes of the melody should be played. So methylation is one mechanism of epigenetics. It can change in any gene during cell development, And the changes that happen for epigenetics can be as harmful as a mutation. So the phenotype can be as strong for an epigenetic change as they can be for a DNA mutation. And methylation is one way that an organism can adapt to its environment. So this is why it's interesting in terms of measuring multiple exposures. The methylation state in a person will actually respond to the exposures that they have had during their life and possibly very early on in their life. So these are the goals for Circle in terms of DNA methylation. We want to establish the methylation patterns that exist during normal B cell development. We want to identify methylation patterns in leukemia and compare those patterns to the normal pattern that we see in B cells. We want to discover also methylation patterns that are specific to environmental factors. So that could be a single environmental factor or multiple environmental factors. And then we want to assess these methylation patterns in the archived newborn blood that we've collected from the cases and controls. So this Venn diagram sort of illustrates the goal. There are a large universe of possible sites for methylation in the green. We're going to first identify the locations where genes are methylated in normal B cell development in the blue here. And then we're going to compare that to the sites where there's methylation in leukemia. And the discordant pairs, the discordant genes, are going to be the ones that might be relevant to leukemia etiology. Likewise, we're going to look at this group of loci where genes are methylated in response to environmental factors. And we're going to try to see in this sweet spot here, what are the genes that are methylated in response to an environmental factor that might also be relevant to leukemia. So that's sort of the goal. So this is the first step. It's the comparison between the leukemia cells and the normal B cells. So in some cases, the leukemia cells were more methylated than the normal B cells at 27,000 loci and 5,000 genes. In other cases, the leukemia cells were less methylated than the normal B cells at 25,000 loci and 10,000 genes. And the plot just shows that. This is the leukemia cells that are less methylated. These are leukemia loci in leukemia cells that were overall more methylated than the normal cells. And we can identify in the Venn diagram the genes that were more or less methylated. So some were less methylated in leukemia cells than in normal cells. Some were more methylated. And there's this group here where some areas of the genes were more methylated and other areas were less methylated. Okay, so then what do we want to do with the environmental factors? Well, if there are 4,772 genes that are more methylated in leukemia cells than normal cells, we're going to also identify the number of genes that are more methylated in children that are exposed to some environmental factor than in the children who weren't exposed to that factor. And then we're going to see how much overlap there is. And the methylation states at these genes where there's overlap are going to represent potential biomarkers of exposure to an environmental factor, something like cigarette smoking, but they're also biomarkers of early effect that may be relevant to leukemia. So they're both biomarkers of exposure and effect. And then to bring it back around to the multiple exposures and cumulative impact theme, we could also look at the intersection between two different exposures and then the discordant pairs of the leukemia normal cells. So we could look at the genes that are methylated by PCBs and the genes that are methylated by cigarette smoking, and then see again this potentially smaller sweet spot that overlaps with the genes that are relevant to leukemia and the methylation states that are relevant to leukemia. So then the methylation states in these genes would represent potential biomarkers of multiple environmental factors and biomarkers of early cumulative effects. So the last section is a discussion of the unique incidence rate of childhood leukemia in the Hispanic population in California. And again, I'm sort of viewing this as an opportunity to assess cumulative impacts of multiple exposures and genetic susceptibility. So this is a plot of the incidence of acute lymphocytic leukemia in California by age at diagnosis and race ethnicity. ALL is the most common of the childhood leukemia types. So in the colors there are in orange, Hispanic, blue is non-Hispanic white, yellow is Asian Pacific Islander, and green is non-Hispanic black. So you can see that for each of the category, the incidence rates are highest for Hispanics. And this is from data from the California Cancer Registry. But it's not a phenomenon that's unique to California. In Florida and Texas, Hispanic ALL incidence rates are also higher than non-Hispanic white incidence rates. And there's some suggestion that potentially Latin American countries in general might also have higher rates of ALL. But that is not something that's totally consistent in all studies. And a potentially more troubling aspect is that not only do Hispanics have a higher incidence rate of ALL, but the rate is also increasing over time. So this is, again, a plot of ALL incidence on the vertical axis, and on the horizontal axis is time. And you can see that in the yellow is the Hispanic group for men, or boys, and girls. And you can see that there's a 1.3% per year increase from 1988 to 2005 for Hispanic girls, and a 2.2% increase per year for Hispanic girls, and 1.3% for Hispanic boys. So this is interesting, but our study isn't really set up to explain why there's a difference. So traditionally we look at the risk of leukemia associated with some environmental factor, and then we use Hispanic ethnicity as a way to stratify the analysis. So we do see that for certain environmental factors, there's a difference in the risk for Hispanics and non-Hispanic whites. So these are 5 factors for which we've identified a differential risk of leukemia associated with a certain environmental factor. But what are really the underlying causes of that difference? Well, one potential answer is a difference in environmental exposures that Hispanics might have. So some things that we've seen in our own study are differential PBDE exposures in settled dust. So this plot shows one of the major PBDE congeners, BD99, in residential dust samples from our study. And then on the horizontal axis, you have non-Hispanic white, non-Hispanic Asian, Hispanic, and then non-Hispanic other races. So you can see, for one thing, that the Hispanic proportion of our population has higher levels of PBDs than the non-Hispanic whites. And then also that this small group of non-Hispanic other races also has very high PBD levels in their dust. But aside from PBDs, there are also different occupational exposures in the Hispanic population. So there are more parents employed as farm or ranch workers. And there are also some differences in the family in terms of the birth order. The use of daycare in the Hispanic population is different. So there's less daycare use in our Hispanic population in the study than there is in the non-Hispanic white population. And the family size is also larger for the Hispanic population than for the non-Hispanic white population in our study. And aside from environmental exposures, there's also potentially different genetic sensitivity in the Hispanic population. So these are 3 of the top hits from the European Genome-Wide Association studies of childhood leukemia. So this is based on a population of European ancestry. These 3 genes and SNPs popped up as being important determinants of leukemia risk. And when we do our own Genome-Wide Association study with the Hispanic population in our study, we see that there's a different risk allele frequency for Hispanics and non-Hispanic whites in our population. So in these risky alleles, which we know are associated with childhood leukemia, in some cases, the Hispanic population has higher allele frequency than the non-Hispanic white population. So potentially that's indicating some increased genetic sensitivity that the Hispanic population has at these specific loci. But this is all sort of, again, piecemeal, where we're looking at certain genes and certain environmental exposures, but we don't have any way of bringing it all together and trying to attribute the difference in leukemia incidence rates between Hispanics and non-Hispanic whites to some set of factors. So what is the explanation for the increased risk? Well, we don't know. There are these unique aspects of the Hispanic population in our study. They have different environmental exposures and different allele frequencies. But we need some sort of systematic strategy to attribute the differential risk to a group of risk factors that might be environmental risk factors or environmental and genetic risk factors. So the question is, do multiple environmental and genetic factors have some sort of synergistic effect that's increasing the incidence rate in the Hispanic population? So to conclude, our study has extensive data on environmental exposures in a large sample size. So it's ideal for systematic analysis of multiple exposures and trying to assess cumulative impacts. But it's not something that we've done yet. We haven't looked at combining joint effects of exposures from especially different classes of chemicals, or also from the joint effect of chemical exposure with social determinants of exposure. So the combined effect of multiple environmental exposures and genetic factors should be considered to explain the risk of ALL in general, but it might be interesting also to look at trying to explain the incidence rate of ALL for the Hispanic population in our study. And the CIRCLE grant will hopefully produce some new biomarkers of cumulative effect of multiple exposures.